Next up is Brad Rogers, and he'll be just discussing gradient.
Good afternoon. My name is Dr. Brad Rogers, and I'm really excited to share with you a new technology that I've helped develop called a temperature gradient device. And I want to show you how this technology can be used as a stability screening platform to predict the shelf life of biological drugs. Biologics, like monoclonal antibodies, are protein-based drugs that represent a growing percentage of revenue for the pharmaceutical industry. These drugs have revolutionized the treatments for diseases ranging from cancer to arthritis. However, as the industry moves toward formulating biologics into small volume shots, shelf life has become a major concern. In fact, it takes about $2.5 billion to bring one drug to market, and a significant contributor to that bottleneck is choosing a drug, the most stable drug, from a library of candidates. And this is because the assays that are used to predict stability, they're slow, qualitative, and not representative of the intended storage conditions. So our solution to this problem is a technology called a temperature gradient device. And by incorporating this patent-pending technology into a stability screening platform, scientists can quickly measure parameters that predict stability. And if you compare our platform to other strategies that are currently employed, our platform is faster, and we obtain multiple metrics for stability. Moreover, our assay measures stability or instability at low temperature, unlike temperature-stressed chromatography, and our assay is performed at high concentration, unlike our closest competitor, Prostabilis. Now, our business model is simple. We will develop and sell an automated platform for about $100,000. And the continued use of this product will require disposable consumables, like disposable sample chips. And there are about 850 biotechs across the world making biologics, and we expect that each one of these companies could be a potential customer, buying 1 to 2 instruments to test the stability of their products. And in fact, we've raised nearly $1 million through collaborations with AstraZeneca and Sanofi to optimize this assay. Moreover, just earlier this year, we were awarded a $250,000 grant from the National Science Foundation to develop our prototype and continue our customer discovery through the I-Core program. The team of scientists and engineers at Penn State are shown in the top left. They will help me build this prototype, and our collaborators in the pharmaceutical industry are shown in the top right, who will ultimately represent our target customer. So looking forward, in 2020, we're going to use that NSF funding to build our prototype, and then by 2021, launch sales to our collaborators and other early buyers, and then by 2022, ramp up production so that we can launch sales to dozens of biotechs across the world. With that, I'd like to thank you and open the floor for questions.
I have a question. Have you validated your technology with known biologics and shown that the shelf life that you predict is actually what it is with marketed biologics?
This is a great question. We're actually underway with a collaboration with Sanofi now. They've given us 7 different commercial antibodies. That material is worth hundreds of thousands of dollars, and we're making our measurements and we're going to correlate it with stability measurements they have for long-term stability. So we're underway with that validation.
Go ahead. I was just going to say, ask if you had any hard data yet on.
We do, yeah. So we've actually started those measurements. We're waiting. It's a blind study, so we don't know what the shelf life is, but we've already started to acquire data for that set of antibodies they provided us.
So you're doing measurements now, but you say you're going to develop a prototype next year. So what is it? What are you doing now? What do you have now? And how does that compare to what is a prototype for, I guess, a minimally viable product?
Yeah, that's a great question. Right now, I like to think or describe our instrument as a garage-style version. It requires some expertise to run the measurements. So our goal over the next year is to make an automated platform. So that's hardware and software that makes this measurement very easy to make in the biopharmaceutical workflow.
And how much of that is hardware and how much of that is software? What's the difficult problem that has to be solved in the next year?
For us, I think it's illustrated with our team here. We have great expertise from the hardware. We have a good handle on what we want to do. Software is something that we're exploring. The analysis is done by hand at the moment, and we know that we need an automated software to streamline the process and extract these parameters.
And is this just analysis software, or is this control software? Is this sort of an interactive, real-time system?
Yes.
What is that?
It's two parts. The first part, like you mentioned, is software to drivers to run the instrument and perform the measurement. And the second part is image analysis to extract these stability parameters.
So can you explain the razor blade analogy a little bit more? I understood the $100,000 platform, the initial platform. What's the razor blade component of that?
So yes, the razor is the instrument, this platform to make the temperature gradient measurements, and the blades are the disposable sample chips. So here I've illustrated a capillary tube which would contain a drug of interest. So we would develop customized microfluidic chips that would work with our instrument. So that's the first blade, and also software updates.
When you're working with pharmaceutical companies, have they defined all the parameters that they're going to need that are important for them, for you to measure to define stability, so that you're making sure you hit every one?
Right. So our assay can measure a number of metrics, and many of these are new. So we're making those measurements, and we're going to compare them with the battery of assays that are currently employed. So in that process, we hope to find out which of these metrics are most useful for predicting shelf life.
And some are required by the FDA, specific ones, so that would be something you'd want to probably incorporate in there. And so your goal then really will be to eventually have this and sell it to companies for their initial screening so that they can more rapidly identify the most stable product. Is that correct?
That's correct, yeah. Preclinical identification of stable candidates. Moreover, once they find a stable candidate, they change the formulation to add a second layer of protection. This assay would also be very useful.
So would this be focused more on new drug development, or would I be able to extend the shelf life of existing drugs if I was a big pharma company?
Yeah, so I guess that touches on the previous question. Our focus has been on identifying stable candidates, but yes, for a candidate that's already down the pipeline, you can add a second layer of protection by changing solution conditions, and our parameters should be provide handles for how stable or how long the shelf life will be for these drugs.
Would that require additional FDA certification, or would it be because I'm using existing elements within certain parameters?
Yeah, this is something that we've been thinking about recently. We think since we're targeting preclinical research and development, that the FDA regulations will be somewhat easier. But if this assay is incorporated into the workflow, then in the quality control steps, then I think that we're going to have to think more seriously about FDA regulations for how precise and accurate these measurements need to be.
Okay, thank you. That was the final question.
Thank you.